Hello everyone, my name is Akhil and I am here to present a PPT on my project, Graph-based detections of mutations in DNA sequences. Coming to the abstract, DNA mutations are important to study due to their role in genetic disorders and evolution. Traditional mutation detection methods rely on linear genome references, which can introduce bias and overlook complex variations. Our approach uses graph structures to detect and visualize mutations, providing a more flexible and comprehensive way to compare DNA sequences. We implement a Python solution that constructs graphs for given sequences, silence mutations, and calculate the similarity scores between them. Coming to the content side, first we'll see an introduction to mutation detection, then we'll move to the project objectives, then we'll give a brief information about the background of DNA and graph theory, then we'll move to methods and implementation, then there's a video on the demo of a code and graph visualization, and we'll discuss about the current progress and pending future works, then we'll see about the differences. To start with the basics, DNA is made of four letters named A, T, C, and G. The mutation just means something in that sequence has changed. It could be a tiny one like base or something bigger like a whole chunk being flipped or deleted. These changes can lead to diseases or something just make us all a little different. So figuring out where and how DNA mutates is super important, especially in medicine and research. The need for graph-based approaches. Most traditional tools like BLAST and BWA use linear genome references, while effective to some extent. They often miss out structural variants and introduce reference bias. Graph-based models allow us to represent multiple parts or versions of a genome, handling the structural variants more effectively. These models are also scalable and suitable for high throughput sequencing data, making them a modern alternative to older approaches. Coming to the objective, our project mainly focuses on these five objectives. Firstly, detect mutations using graph structures, then visualize DNA sequencer directed graph, next highlighted mutation portions clearly, next calculate the similarity between the sequences, next compare the graph based and traditional approaches. Before moving to the methodology, let's briefly go over the types of mutations. SNPs, single base changes, insertions or deletions, also called as indels. It means the addition or removal of bases. Structural variants, larger changes like duplications, inversions, or translocations. Mutations can be inherited or caused by external factors like UV radiation or replication errors. Introduction to graph theory. A graph is just a bunch of nodes connected by edges. In our case, each node can represent a DNA base or a group of bases. The edges show how they are connected. Like in actual DNA sequence, we use two types of graphs: deep Bruin graphs, which are great for reconstructing sequences for smaller parts, and variation graphs, which help us compare differences between sequences. Coming to graph applications in genomics, graph theory has already been applied in many areas of genomics, such as genome assembly. Deep, in genome assembly, deep Bruin graphs help rebuild genomes from short DNA reads. In variant calling, variation graphs help detect mutations more precisely. In pangenomics, graphs allow us to represent genomic diversity across multiple individual or populations. Coming to the methodology, firstly two DNAs are given. One is reference DNA and another one is test DNA. Our objective is to find the different mutations, means the differences between the test and reference DNA. Firstly, we'll compare each nucleotide one by one and we'll identify a difference and we'll mark it with different color. Coming to the second step, we convert the sequence into direct graphs. Each node represents a base along with its positions, example 0 for A, 1 for T, 2 for C, etc. It just connect consecutive nodes to show sequence flow. We create separate graphs for the reference and test sequences. In test graph, each node is labeled as mutated or not based on comparison with the reference. Coming to visualization, using Python libraries like NetworkX and Matplotlib, we can plot the graphs. Reference nodes are shown in blue, test graph nodes are green if unchanged and red if mutated. We can also compute a similarity score based on the percentage of matching two letter subsequence like ATTG, GT. This helps us quantify the degree of similarity between the two sequences. Coming to the progress summary, so far we successfully developed the Python script for mutation detection. Then the visualization part using NetworkX and Matplotlib is also completed. The tool correctly identifies and marks mutation points, and it also calculates the similarity scores. And we also test with the example DNA sequence, and the results are some accurate. Coming to the challenges in future work, there are a few challenges that we are currently facing. Handling longer DNA sequences require better graph layouts and performance tuning. Large graphs can become hard to interpret visually. We cannot identify the mutated genes that easily in large DNA sequences which are represented in the graph. In the future, we plan to extend this approach to full-length genomic sequences. We should also optimize the algorithm for better speed and scalability, and we should possibly integrate this with other bioinformatic tools. Coming to the summary, we demonstrated a graph-based method to detect and visualize mutations in DNA sequences. This approach overcomes the limitations of linear model, particularly in detecting complex variations. Our Python code implementation works efficiently, and it's also providing clear visuals for the graphs and accurate similarity scores. With further development of this method, it can be applied to real-world bioinformatics and research tasks. Coming to the references, we use the first link to obtain different types of DNA sequences for testing our similarity score and different types of graphs. We use the network documentation to understand more about the libraries like Matplotlib and NetworkX. We also use the third link to understand more about deep neural graphs in the genome assembly. Thank you.